Welcome to our biology class. This is Dr. Ramakrishna, senior biology faculty. Previous video or previous class we can discuss specially the structure of kidney and what about the excretory material, what is the excretory organs we are discussed in a previous video. Now this video we can discuss specially the structure of nephron that will be discussed. Okay. So normally every kidney will be consist nearly above the 1 million. Each and every kidney will be consist nearly 1 million of the nephrons will be present. So what is the nephrons? We know cell is the basic unit of the living organism. The number of cells will be together. We have the tissues, organs and organ systems. At the same time here our kidney also consists 1 million of the nephrons will be together. 1 million of nephrons will be together. Our kidney will be formation. So the nephron is the structural and functional units of our kidney. Okay. So now we can discuss about the structure of the nephron. But what about the nephron? The nephron, it will be consist of the two regions. That is the Bowmans. First one is the malphizian body and the second one is the renal tubule. Also nothing but a tubular, renal tubule. So but first of all we can discuss about the Bowmans capsule. But what is the Bowmans capsule? Specially Bowmans capsule means it is a double layer, double membrane, one of the cup-like structure or one of the, for example, a capsule or one of the bowl-shaped structure, that is nothing but a Bowmans capsule. In the Bowmans capsule, we have the, look at here, one is the efferent arteriole, one is the efferent arteriole. Especially the efferent arteriole means nothing but it is a renal vein. Means that will be collecting, that is collecting the deoxygenated blood and that will be leads into the Bowmans capsule. And the efferent arteriole means that is nothing but it is the after purification or after filtration that will be collecting the blood from the kidney. Okay, so a double membranous or double layer, one of the bowl or one of the cup shaped structure is the Bowmans capsule. Inside the Bowmans capsule, the efferent arteriole, it will be divided. The efferent arteriole, it will be divided into number of branches. Number of branches will be present and such as the branches also will be having the small capillaries like structures. The small capillaries like structures will be there. At the same time, in between the capillaries, they have the minute spaces will be present. Such as the minute spaces will be consist, a specialized cells will be present in the Bowmans capsule. A specialized cells, a specialized one of the cells will be present. These specialized cells is normally we can say podocytes. These are normally we can say the podocytes. The efferent arteriole, it will be divided into, it will be consist number of branches. The branches also will be consist number of capillaries. The in between the capillaries, they have the minute pores will be present, such as the minute pores will be consist, specialized cells are there. These are the podocytes, right? At the same time, the capillaries also will be consist minute or some of the small pores will be present. That is normally we can say slit pores. Slit pores. But what is the importance of the slit pores? Through the blood will be enter into the Bowmans capsule. Into the in presence of the slit pores, they will be perform the filtration. They will be perform the filtration. So this is normally we can say glomerular region. This is normally we can say glomerular region. So in the glomerular region, the blood will be performing the purification. So that normally we can say glomerular filtration. Glomerular filtration. Glomerular filtration. Also nothing but a ultra. Also nothing but a ultra filtration. Glomerular filtration. Also nothing but a ultra filtration that will be performed in our Bowmans capsule. So Bowmans capsule means it is a double layer, a double membrane, one of the cup-like or one of the capsule, one of the bowl-shaped structure is the Bowmans capsule. Inside that Bowmans capsule, especially the afferent arteriole will be entered. The afferent arteriole will be divided into number of capillaries will be there. In presence of the blood capillaries, they have the small minute spaces are there. That is the they will be filled by one of the specialized cells, that is the podocytes. And along at the same time, the capillaries also will be consist of small pores. Through the pores only, the blood will be perform the filtration. So that is normal. We can say glomerular filtration, also nothing but ultrafiltration. Actually, in our urine formation, the formation of urine also, we have the 3 stages will be there. One is the glomerular filtration. The second one is the selective reabsorption. Third one is the tubular secretion. So the glomerular filtration means totally the blood will be performing the filtration except plasma proteins. Except the plasma proteins, remaining all components will be performing the filtration. So that is the glomerular filtration that will be present in our Malpighian tube, Malpighian body, also nothing but a Bowmans capsule. After completion of the Bowmans capsule, we have the renal tubule, also nothing but a tubular like structure will be there. So this is the normal, we can say renal tubule, also nothing but a tubular like structures will be there. This is normal, we can say renal tubule. But what is the renal tubule, sir? Especially in the Bowmans capsule, the afferent arteriole, it can be leads into the Bowmans capsule. After completion of the purification, the blood, it will be passes into the one of the highly coiled tubular like structure. So that is normally we can say here we have the one of the tubular, highly coiled tubular like structures will be there. But what is the tubular like structure? Here, look at here. After Bowmans capsule, there are the number of coils and also one of the descending. Descending means nothing but the downwards. So this is the one of the descending limb of this is the one of the descending limb, and here we have the one of the ascending limb. So as descending limb and ascending limb. So the ascending and descending limbs both will be together a U-shaped. Both will be together here. A U-shaped structure will be formed. A U-shaped structure will be formation. So this is the U-shaped structure. This U-shaped structure normally we can say Henle's loop. Normally we can say Henle's loop. So Henle's loop also normally we can say hairpin. Hairpin also nothing but a U-shaped. A U-shaped structure will be formation. That is normal we can say Henle's loop. The Henle's loop will be having the 2 limbs. One is the descending limb and also one is the ascending limb. Then after ascending, then afterwards what will happen? One of the highly coiled, one of the structure will be formation. This is nothing but a collecting duct. This is nothing but a collecting duct will be there. This is the collecting duct. And at the same time, after entering into the filtration, the efferent arteriole, then afterwards the purification will be completed. Then will be elimination. The blood, purified blood will be passing through the efferent arteriole. That is the efferent arteriole. The efferent arteriole also will be formation just like a PCT. What is the meaning of the PCT? Means proximal converted tubule. Proximal converted tubule, and here one of the highly coiled structure will be there. That is normally we can say DCT. What is the meaning of the DCT? Means a distal converted tubule. Distal converted tubule, and along with them we have the one of the another structure also will be present in the renal tubule. That is normally we can say vasa recta. Vasa recta. But what is the vasa recta? Special lead. The efferent arteriole, it can be leads into the Bowmans capsule. They will be divided into number of capillaries or number of tubules. Such as the capillaries and tubules, will be formation one of the capillaries like structure. Such as the capillaries also is conversion to a tubular like structure. Such as the tubular like structures also will be surrounded by the tubular. This is the peritubular. The peritubular structures also will be surrounded by the Henle's loop. That is nothing but a vasa recta. Vasa recta means The peritubular membrane or peritubular structures also will be surrounded our loop of Henle. That nothing but a Henle's loop, that is nothing but a vasa recta. So that is the generally, and at the same time in our kidney, actually we know the structurally and functional units of the nephrons is the structural and functional units. Such as the nephrons also, we have the two types of the nephrons are there. There are the two types of the nephrons. The nephrons, two types. One is the corticular and the second one is the medullary. The nephrons also will be bifurcate or consist of, there are two types. One is the corticular and the second one is the medullary nephrons. But what is the difference between the corticular nephrons and also medullary nephrons? Especially we know normally our kidney, there consist of the two regions. Outer is the cortex, inner is the medulla. Outer is the cortex and inner is the medulla. So especially some of the nephrons, the Henle's loop, some of the nephrons, they are consist of the Henle's. Normally we know the Henle's loop will be there. Some of the nephrons of Henle's loop is very short. So the very short of the nephrons are their leads, they will be present into the cortex region. They are unable to enter into the medulla. So especially some of the nephrons, the U-shaped Henle's loop. The Henle's loops are very short. So they can lead into the only cortex region, such as the cortex region. Short Henle's loops nephrons are normally we can say corticular nephrons, and some of the nephrons are they have the whatever the specially a longitudinal U-shaped Henle's loops will be present. They will be leads into the up to the medullary region. Such as the nephrons are normally we can say medullary nephrons. Also normal we can say juxtamedullary nephrons. The juxtamedullary nephrons, they are consist of the longitudinal Henle's loops will be there. They can from the cortex region to they can leads into the medullary region, such as the nephrons is normally we can say juxtamedullary nephrons. These the juxtamedullary nephrons are will be performing the highly coiled and also longitudinal Henle's loops will be there. So this is the normally structure of our nephron. Then afterwards we can discuss especially the formation of urine. But what is the formation of urine, sir? Generally, the formation of urine, we have there are three regions will be there. There are the three methods are there. Especially, what is that? First of all, glomerular filtration, selective reabsorption, tubular secretion. There are the three methods are present in our formation of urine. Very simple actually. The formation of urine consists of three methods. One is the glomerular filtration. The second one is the selective reabsorption. Third one is the tubular secretion. But what is the glomerular filtration? Means we know, especially the glomerular filtration, it will be performed in our Bowmans capsule. Particularly, we know the afferent arteriole, it will be consist a number of branches, 5 branches will be there. So along with them, they are consist the specialized epithelial cells, that is the podocytes. At the same time, the capillaries also will be consist small pores will be there, that is the slit pores. So through the podocytes and slit pores, the blood will be performing the filtration. So except plasma proteins. What is the plasma proteins? Normally we know albumins, globulin, fibrinogen. So such as the albumin, globulins, these are the common we can say plasma proteins. Except plasma proteins, remaining all permeable and impermeable components also will be performing the filtration in the glomerular region. So the total except the plasma proteins, remaining all components of our blood, they will be performing the purification in our Bowmans capsule. That is normally we can say glomerular filtration. Glomerular filtration, also nothing but a ultrafiltration. After completion of the glomerular filtration, in our body normally we have the approximately 180 liters. 180 liters of the urine or primary urine will be formed. But 180 liters of the urine we are unable to excrete. So out of the 180 liters of the urine, remaining 178.5 liters. 178.5 liters of the urine that will be processed through the the PCT and DCT and also loop of Henle. Whenever they will be processed into the renal tubule, they will be reabsorption. That is nothing but a selective reabsorption. But what is the selective reabsorption? Means especially after formation of the glomerular filtration, there approximately 180 liters of the primary urine will be formed. That primary urine also will be consist of the plasma proteins and some of the useful minerals, and also some of the elements also will be present. For example, sodium, potassium, calcium, magnesium, such as the components, and also some of the useful minerals also will be present in this 180 liters of the urine. So that's why whenever they will be passing through the renal tubule, also nothing but a descending limb and also ascending limb, and a PCT and DCT, whenever it will be moving. So what will be happen? The selective reabsorption. What is the selective reabsorption? For example, before in our home also, for example, we have some garbage. So before elimination, before throwing that, one of the useful components or any useful materials also will be recollect. Here also, after formation of the glomerular filtration, whenever they will be passing in the descending limb and also ascending limb, the useful components like some of the sodium, chlorides and water, and also some of the useful elements like sodium, potassium, calcium, magnesium, and also some of the chlorides and also sodium and also water, they will be absorption through the collecting duct. That is nothing but a selective reabsorption. So 178 liters of the urine will be formation in a, but 178.5 liters are reabsorption. Then afterwards, the tubular secretion. The tubular secretion means after completion of the elimination, after completion of the reabsorption, the wastage products will be elimination from our body. That is nothing but a tubular secretion. Approximately 1.5 liters. Approximately 1.5 liters of the urine we can excrete in 24 hours. Not in 1 hour. In 24 hours we can excrete perfectly, means 1.5 liters of urine we can excrete in 24 hours. For example, one of the person, he will be or she will be excrete excess amount of above 1.5 liters, means normally 2 liters, 2.5 liters, means they are suffering some of the problem. Why? Because normally we can in 24 hours we can excrete that generally 1.5 liters of the urine only we can excrete, but due to some of the hormonal imbalancing or due to some of the hormone deficiency, the person will be excrete the maximum percentage of the urine. So that's why we are facing some of the troubles. That is nothing but a diabetes. The diabetes also, we have the two types. One is the diabetes insipidus and diabetes mellitus. Diabetes insipidus and diabetes mellitus. What is the diabetes insipidus? What is the diabetes mellitus? For example, the diabetes mellitus means the diabetes mellitus, one of the disease, it will be deficiency of the insulin. The deficiency of the insulin. For example, in our human being, we have the pancreas, one of the gland will be there. The pancreas, it will be secreted. There are the two types of the hormones. One is the insulin, the second one is the glucagon. But what is the importance of the insulin? The excess amount of the glucose molecules. The excess amount of the glucose molecules is conversion to glucose and store in our body. That is the function of insulin. But due to some abnormalities, due to some of the heredity, or due to some of the different problems, what will happen? The insulin secretion will be low, or sometimes it will be absent. So the deficiency of the insulin. So what will be happen? The excess amount of the glucose molecules, it is unable to conversion to glycogen. So the excess amount of the glucose molecules, it will be elimination through the urine. That is nothing but a diabetes mellitus. Normally we can say sugar, right? So that is the diabetes mellitus. But what about the diabetes insipidus, sir? The diabetes insipidus, it is also one of the excess elimination of the urine. Normally we can elimination 1.5 liters, but due to the absence of one of the hormone, that is the ADH, anti-diuretic hormone. Anti-diuretic hormone, that is normally we can say vasopressin. But what is the importance of the vasopressin? The vasopressin also nothing but ADH, it will be reduced. It will be reduce the elimination of the urine content, it will be reduced. But unfortunately, the pituitary gland, actually the ADH, one of the hormone, it will be secreted by the pituitary gland. So the ADH hormone is abnormality or absent or deficiency. So what will happen? The person also, they wanted to elimination the maximum percentage of the urine. So that is commonly we can say diabetes insipidus. But what is the difference between the diabetes insipidus and diabetes mellitus? Both also excrete the maximum percentage of the elimination of the urine. But the diabetes mellitus person excrete in that urine, it will be having the glucose. At the same time, the deficiency of ADH, the person excretion of that urine, the glucose is absent. So that is the difference. Glucose is absent means that is diabetes insipidus. Glucose is present, that is normally we can say diabetes mellitus. Okay. So after completion of that, the purification, reabsorption, then after it will be leads into the ureter, and through the ureter will be leads into the urinary bladder. So we know the urinary bladder, will be stored, it will be having the capacity up to 400 to 700 milliliters of the urine. So once it will be filled by the urine, so what will happen? One of the stretch impulses, it will be leads into our brain. We wanted to excrete the urine, such as the feeling will be happy. So we are having, so that time we can elimination or excretion, especially the urethra. The urethra will be consist, one of the specialized sphincters will be there. The sphincters are performing the contraction, we can elimination of the urine. That is normally we can say micturition. The process is nothing but a micturition. So especially the micturition, it is a voluntary reaction in our adults, but infants it is the involuntary. Right? So involuntary means particularly, especially the below 1 year, below 2 years, they are the involuntary means they are unable to control the urine. So that's why, for example, whenever we are taking that small infants, automatically they will be excrete their urine. So that is the involuntary in the infants, but adults it is the voluntary. So this is the process of our excretory system, especially the Bowmans, particularly the structure of nephron, and at the same time we are discussing the formation of urine. So this is our excretory system. Okay, I hope that you are following this class. So you can subscribe and also like and share to your friends. Thank you.